Hello students and welcome to the lecture on Biosynthesis of fatty acids. The main objectives of today's lecture are to give a general description of fatty acids, to explain saturated and unsaturated fatty acids, to give a general idea of essential and non-essential fatty acids, to explain in detail the biosynthesis of fatty acids, to give a brief idea of regulation of fatty acid synthesis. To begin with, let me first of all give a brief introduction about fatty acids. Fatty acids are a class of compounds containing a long hydrophobic hydrocarbon chain and a terminal carboxylic group. Fatty acid synthesis in animals occurs in cytosol and in plant cells in chloroplast stroma. Fatty acid synthesis is the creation of fatty acids from acetyl coenzyme A and NADPH through the action of enzymes called fatty acid synthesis. Fatty acids are the simplest form of lipids and serve as a constituents in a large number of complex lipids. Fatty acids are long-chain hydrocarbons, 4 to 36 carbons long, with one carboxylic group. Fatty acids are amphipathic by nature. Carboxylic group of a fatty acid is hydrophilic, polar or water-soluble, while the rest of the hydrocarbon chain is non-polar, hydrophobic or water-insoluble. Polar end sticks to water so that fatty acids form a monomolecular layer over it. The water-soluble end of the fatty acid also reduces surface tension and increases the wetting or cleaning power of water. Fatty acids have four major physiological roles. Number one, fatty acids are building blocks of phospholipids and glycolipids. These amphipathic molecules are important components of biological membranes. Number second, many proteins are modified by the covalent attachment of fatty acids, which targets them to membrane locations. Number third, fatty acids are fuel molecules. They are stored as which are uncharged esters of fatty acids with glycerol. Fatty acids mobilized from triacyl glycerols are oxidized to meet the energy needs of a cell or organism. Number four, fatty acid derivatives serve as hormones and intracellular messengers. Now, let us discuss the saturated and unsaturated fatty acids. Saturated fatty acids have no double bonds in the chain. Examples of predominant saturated fatty acids are lauric acid, myristic acid, palmitic acid, and others. Unsaturated fatty acids have one or more double bonds and are called monounsaturated or polyunsaturated fatty acids respectively. The double bonds in naturally occurring fatty acids are generally in a cis as opposed to a trans configuration. Unsaturated fatty acids have lower melting point than the saturated fatty acids. Most plant lipids have unsaturated fatty acids, while most animal lipids have saturated fatty acids. Aquatic animals of cold waters, however, possess unsaturated fatty acids. A saturated acyl group produced by this set of reactions becomes the substrate for subsequent condensation with an activated malonyl group. With each passage through the cycle, the fatty acyl chain is extended by 2 carbons. When the chain length reaches 16 carbons, the product palmitate leaves the cycle. Carbons C16 and C15 of the palmitate are derived from the methyl and carboxyl carbon atoms respectively of an acetyl coenzyme A used directly to prime the system at the outset. The rest of the carbon atoms in the chain are derived from acetyl coenzyme A via malonyl coenzyme A. Both the electron-carrying cofactors and the activating groups in the reductive anabolic sequence differ from those in the oxidative catabolic process. In oxidation, NAD positive and FAD serve as electron acceptors and the activating group is the thiol group of coenzyme A. By contrast, the reducing agent in the synthetic sequence is NADPH and the activating groups are two different enzyme-bound thiol groups. All the reactions in the synthetic process are catalyzed by a multi-enzyme complex, fatty acid synthase. Although the details of enzyme structure differ in prokaryotes such as E. coli and in eukaryotes, the 4-step process of fatty acid synthesis is the same in all organisms. Dear students, let us now discuss the essential and non-essential fatty acids. Based on whether the body can produce fatty acids can be classified into essential and non-essential fatty acids. The main difference between the two is that essential fatty acids cannot be produced by the body and have to be consumed through food or dietary supplements. Non-essential fatty acids can be produced by the body, although they can still be ingested from some of the food that we eat. Essential fatty acids. Basically, for humans, there are only two kinds of essential fatty acids. These are the alpha-linoleic acid, which is the kind of omega-3 fatty acid, and the linoleic acid, which is a type of omega-6 fatty acid. There is a third category of essential fatty acids which are actually known as conditional essential fatty acids because they become essential only on certain developmental or illness conditions. Examples of conditional essential fatty acids are the gamma-linoleic acid, which is a kind of omega-6 fatty acid, and docosahexaenoic acid, which is an omega-3 fatty acid. The omega-9 fatty acids are considered to be non-essential fatty acids because they can be produced using other fatty acids and carbohydrates. The balance and ratio between the three kinds of omega fatty acids is important. Many factors influence the need for the omega acids and affect the ratio in which they should be consumed. Generally, the amount of omega-3 should be higher than that of the omega-6. Age and gender are the most important factors. Omega-6 is found in vegetable oil, and it is believed that this particular type of omega fatty acid helps with the regulation of blood flow and levels of blood cholesterol. Omega-3 has an anti-inflammatory effect, especially beneficial to the heart when inflammation of the vessel walls cause atherosclerosis. It's mainly found in fish oils such as tuna and mackerel. Certain nuts contain fatty acids, especially almonds and walnuts, but it is important to eat them in moderation. Dark leafy vegetables like spinach and kale and flaxseed also contain omega-3 fatty acids. Now, non-essential fatty acids. Some non-essential fatty acids are actually important for the body. Of course, the saturated fats are considered to be non-essential fatty acids because they are not required by the body and instead may cause harm. Trans fatty acids, although these are a kind of unsaturated fats, are also non-essential fatty acids and should be avoided in the same manner that we should avoid saturated fats because of findings that increased consumption of trans fats is proportional to an increase in the risk of coronary heart disease. It's also important in supplements when used as development enhancements. Meanwhile, there are non-essential fatty acids which are simply classified as such because the human body can synthesize them from other nutrients like carbohydrates and other unsaturated fatty acids. These are the omega-9 fatty acids which can lower blood cholesterol and control blood sugar. Now, let us move to the main part of today's lecture, that is the fatty acid synthesis. De novo synthesis of fatty acids occurs predominantly in liver, kidney, adipose tissue, and lactating mammary glands. The enzyme machinery for fatty acid production is located in the cytosomal fraction of the cell. Fatty acid biosynthesis in the cytosol requires a sufficient concentration of NADPH and acetyl coenzyme A. NADPH is generated in the cytosol by the pentose phosphate pathway and by the malic enzyme which oxidizes malate into pyruvate and carbon dioxide, generating NADPH. The acetyl coenzyme A formed by amino acid degradation is insufficient for fatty acid biosynthesis. Acetyl coenzyme A molecules are the building blocks of fatty acid synthesis. There are three principal ways of producing acetyl coenzyme A in the cytosol of the cell. Number 1, amino acid degradation. Number 2, fatty acid oxidation, and number 3, glycolysis. The acetyl coenzyme A molecules need to be activated for fatty acid biosynthesis. The pathway for the biosynthesis of saturated fatty acids is identical in all organisms. A sequence of 7 enzymes catalyzed reactions converts 2 carbon units to 4 carbon units. The first reaction is acetyl coenzyme A is carboxylated to form malonyl coenzyme A by acetyl coenzyme A carboxylase, which is a biotin-containing enzyme. The carboxylation reaction is irreversible and is the first committed step of fatty acid biosynthesis. ATP is used to activate bicarbonate in the form of carboxyphosphate, which leads to the carboxylation of biotin. The activated carbon dioxide group is transferred to acetyl coenzyme A to form malonyl coenzyme A. The reaction is HCO3 plus ATP plus acetyl coenzyme A gives ADP plus inorganic phosphate plus malonyl coenzyme A. Acetyl coenzyme A carboxylase has 3 domains. Number 1, a biotin carboxyl group carrier protein. Number 2, biotin carboxylase which adds carbon dioxide to biotin. And number 3, a transcarboxylase which transfers the carbon dioxide group from biotin to acetyl coenzyme A to form malonyl coenzyme A. The remaining reactions of fatty acid synthesis are catalyzed by a multifunctional enzyme complex called fatty acid synthase complex. Fatty acid synthase is a polypeptide chain with multiple domains, each with distinct enzyme activities required for fatty acid biosynthesis. It's not a single enzyme, but a whole enzymatic system composed of 2 identical 272-Clu-Dalton multifunctional polypeptides in which substrates are handed from one functional domain to next. Its main function is to catalyze the synthesis of palmate from acetyl coenzyme A and malonyl coenzyme A in presence of NADPH. There are 2 types of fatty acid synthesis. Type 1, found in both mammals and fungi. Type 1 fatty acid synthase system is also found in corynebacteria, mycobacteria and nocardia. In these bacteria, FSC1 system produces palmitic acid and cooperates with the FSC2 system to produce a greater diversity of lipid products. Type 2 is found in bacteria and archaea and is characterized by the use of discrete monofunctional enzymes for fatty acid synthesis. In eukaryotic cells, including humans, the fatty acid synthase exists as a dimer with 2 identical units. Formation of acetyl ACP and malonyl ACP. The 2 carbon fragments of acetyl coenzyme A are transferred to ACP of fatty acid synthase catalyzed by the enzyme acetyl coenzyme A, ACP transacylase. The acetyl unit is then transferred from ACP to cysteine, residue of the enzyme. Thus, ACP site falls vacant. The enzyme malonyl coenzyme A, ACP transacylase, transfers malonate from malonyl coenzyme A to bind to ACP. Other reactions in the formation of fatty acids include step 1, the condensation. The first reaction in the formation of a fatty acid chain is condensation of the activated acetyl and malonyl groups to form acetoacetyl ACP, and acetoacetyl group bound to ACP through the phosphopentathenine thiol group. Simultaneously, a molecule of carbon dioxide is produced. This reaction is catalyzed by beta-ketoacyl ACP synthase. Step 2, reduction of the carbonyl group. The acetoacetyl ACP formed in the condensation step now undergoes reduction of the carbonyl group at carbon 3 to form dehydroxybutyryl ACP. This reaction is catalyzed by ketoacyl ACP reductase and the electron donor is NADPH. Step 3, dehydration. The elements of water are now removed from C2 and C3 of dehydroxybutyryl ACP to yield double bond in the product trans-2-butynyl ACP. The enzyme that catalyzes this dehydration is hydroxyacyl ACP dehydratase. Step 4, reduction of the double bond. Finally, the double bond of the trans-2-butynyl ACP is reduced or saturated to form butyryl ACP by the action of enoyl ACP reductase. Again, NADPH is the electron donor. Synthesis of palmitic acid. The carbon chain attached to ACP is transferred to cysteine residue and the reactions cited above are repeated 6 more times. Each time the fatty acid chain is lengthened by 2 carbon units. At the end of the 7 cycles, fatty acid synthesis is complete and a 16-carbon fully saturated fatty acid palmitate bound to ACP is produced. The enzyme palmityl thioesterase separates palmitate from the fatty acid synthase by a hydrolytic activity in the complex. This completes the synthesis of palmitate. The overall reaction of the fatty acid synthesis is 8 acetyl coenzyme A plus 7 ATP plus 14 NADPH plus 14 H positive gives palmitate plus 8 coenzyme A plus 7 ADP plus 7 inorganic phosphate plus 14 NADP positive plus 6 water molecules. Now, moving on to the last part of today's lecture, that is the regulation of fatty acid biosynthesis. Acetyl coenzyme A is formed into malonyl coenzyme A by acetyl coenzyme A carboxylase. At this point, malonyl coenzyme A is destined to feed into the fatty acid synthesis pathway. Acetyl coenzyme A carboxylase is the point of regulation in saturated straight chain fatty acid synthesis and is subject to both phosphorylation and allosteric regulation. Regulation by phosphorylation occurs mostly in mammals, while allosteric regulation occurs in most organisms. Allosteric control occurs as feedback inhibition by palmitoyl coenzyme A and activation by citrate. When there are high levels of palmitoyl coenzyme A, the final product of saturated fatty acid synthesis, it allosterically inactivates acetyl coenzyme A carboxylase to prevent a build-up of fatty acids in cells. Citrate acts to activate acetyl coenzyme A carboxylase under high levels because high levels indicate that there is enough acetyl coenzyme A to feed into the Krebs cycle and produce energy. High plasma levels of insulin in the blood plasma cause the dephosphorylation of acetyl coenzyme A carboxylase, thus promoting the formation of malonyl coenzyme A from acetyl coenzyme A and consequently the conversion of carbohydrates into fatty acids, while epinephrine and glucagon cause the phosphorylation of this enzyme, inhibiting lipogenesis in favor of fatty acid oxidation via beta oxidation. Students, that's all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.